Very good afternoon to the respected speaker, to our co-organizers and all the participants. We welcome you back to the live webinar series Sustainable Biosolutions 2020. Now, as we begin every session, we would like to give a short introduction about who we are. So, I am Dr. Viraj Gala. I work as the marketing manager for biomol.in. Now, biomol.in, it is India's first and one of the world's leading online portal for laboratory life science and diagnostic products. We have 1 lakh plus products from 150 plus manufacturers. Most of them are Indian manufacturers, and we ship all kinds of laboratory products worldwide to every 3 tier and 4 tier cities. We can provide any product that you need. Now, may I request Dr. U. Kaushik Kumar, Assistant Professor at the Department of Biotechnology, to please introduce Welltech Rangarajan, Dr. Saguntala, R&D Institute of Science and Technology, and please take the session ahead.
Thank you, Dr. Viraj. I welcome you all, everyone who was present for this webinar series. We at the Department of Biotechnology will take Rangarajan, Dr. Sagundala R&D Institute of Science and Technology, Chennai, are happy to bring these webinars in association with that of biomol.in. So before going to that of introduction, just let me introduce our institute. The Welltech Chennai was established in the year 1987 by Dr. R. Rangarajan and Dr. R. Sagundala Rangarajan. The institution is a private deemed to be university and is ranked number 5 in the country by MHRD for innovations. Has a diamond rating from iGauge, 43rd among the Asian university in the Times of India Higher Education World Rankings. Our Department of Biotechnology aims to create and promote upskill graduates with a professional attitude who are industry ready. So I'm very happy to introduce the speaker today's session, Professor Stefan Mikael, a professor of environmental geochemistry at Ecole Polytechnique of University of Orléans, France. He holds a master's in oceanology, a doctorate in environmental chemistry from University of France, former research associate at University of Lancaster, scientist at French Geological Survey. In 2006, Professor Mikael joined the University of Orléans as a full professor. He has developed expertise in environmental biogeochemistry, environmental mineralogy, and ecological restoration. As a co-author, he has more than 130 peer-reviewed articles and 180 presentations at the conferences with somewhere 3200 citations. His H index is 32 in Google Scholar. He is a recipient of prestigious International Association of Advanced Materials Medal in year 2017. Welcome, sir. I now pass on the session to Professor Stephen Mikelo. Thank you.
Okay, dear friends, dear scholars, dear researchers, dear professors, dear students, many thanks again for Welltech and Dr. Manoj for this kind opportunity to present some of my research work. I remember well the good time I had when I spent 2 weeks in Welltech some 2 years ago, teaching with students of Dr. Manoj and also students from Civil. It was a really great time in Welltech in India. And today I would like to introduce some new work about trace element cycling in particular hotspots of the critical zone. I will define later what I mean by critical zone, but it's basically a zone in the Earth's surface that is critical to the life of human beings and other living species, from the top of the watershed to the top of the trees, basically the surface environment we're living in. And I would like to move from theoretical consideration, fundamental research, to more applied bioremediation, phytoremediation, and even zooremediation of contaminated soils. And I quite like geochemistry, but in a broader perspective in relation to life and humans. My research interest is environmental biogeochemistry of trace elements, while meeting new challenges in the field of biogeochemical functioning of terrestrial and aquatic systems within the anthroposphere. And so far, I have carried out multidisciplinary research during my career, focusing on the study of the ecodynamics of trace elements. And I have divided research in 3 axes and on. So after a few words about some background information about our university and research introduction and research institution, I will give you some details about the context of my work, then some research objectives for the presentation of today, then some insight into the material and method we use to carry out these research projects, and then I will jump into the results. And last but not least, I will conclude with some conclusive remarks. Yeah, so we are located in the heart of France, not so far from Paris, like 120 kilometers south of Paris, on the beautiful River Loire in the region Centre, the center of France, in the city of Orléans. You can see a picture of our city, which is divided into 2 parts, north of the River Loire and south of the River Loire, at the bottom of the PowerPoint of the slide. And on the top right-hand side of the picture, you can see the River Loire, which is the longest river in France, spreading from the mountains and the volcanoes of the region of Clermont-Ferrand up to the Atlantic Ocean. And we are right there, south of Paris. Our university is a very old one. It has been established in the Middle Ages and then has stopped for several hundred years and started again in 1968. And I'm teaching there at the Department of Civil Engineering and Environment in our faculty, which is called the Ecole Polytechnique de l'Université d'Orléans. Basically, it is the engineering school of our university. And I do teach environmental science, basically some fundamentals such as geology, surface geology, pedology, hydrogeology, and hydrology. And then my specialties for master students are geochemistry, biogeochemistry, environmental remediation, ecological restoration, and also ecotoxicology. So this is my academic background. And then I would like to introduce our research group and how we link research to teaching. We are very fond of geoenvironmental engineering, and for us, we develop some specific expertise in risk assessment, contamination studies, and reclamation of contaminated lands, together with waste management. And I do believe that environmental geochemistry, together with contaminant hydrogeology, but also environmental microbiology, are essential for the skills developed by our students. So we try to teach in an interdisciplinary way our students, our future engineer of civil and environmental technology. I will move now to the next slide. This is basically how the different skills we try to develop among our students interact. Basically, geochemistry is a basis because in order to assess the processes that will control pollutants, mobility in the environment, such as soap show, biodegradation, even radioactive decay, and also the mobility into the trophic chains of terrestrial and aquatic ecosystems. We need to combine this geochemical approach with soil science and hydrology, even hydrogeology. And modeling is also something we developed. And I would like to introduce further some research work where we combine observation in the field, mesocost experiments in the laboratory, together with numerical modeling with computers in silica. And last but not least, to assess the risk to human health and the environment, we also integrate some microbiology and ecological aspects. And last but not least, and then the loop will be closed, we integrate this in a broader perspective of civil engineering to design remediation strategies based on low-tech approaches, based on nature-inspired solutions, such as bioremediation, phytoremediation, or even zooremediation. This is basically our philosophy. We do work at the Institute of Earth Sciences of our university, which is a joint research center between our university, the French National Research Center CNRS, and also the nearby French Geological Survey, BRGM. And all aspects of Earth and environmental science are covered in this laboratory, but our group basically focuses on biogeochemical processes in continental context, such as watersheds, soils, groundwaters, and the interface between soils and the proximate atmosphere. This is our research group. And this is the approach we try to develop from fundamental biogeochemistry through ecological restoration. And this will be the focus of today, to agroecology and help in improving designing new agricultural solutions. So geochemistry, mineralogy, soil science, and hydrology overlap together with microbiology to understand how the natural environmental work, how we can also understand the mobility and ecotoxicology and ecodynamics of contaminants in contaminated terrestrial ecosystems, and how we can improve and design remediation strategies for these contaminated systems. I will move to next slide. And I will now introduce, can you see all this slide taken from the USGS, the United States Geological Survey, which is a critical zone, the concept of critical zone as defined by Brantley et al. Basically, the critical zone is the Earth's permeable layer that extends from the top of the vegetation to the bottom of the groundwater zones. It interconnects rocks, soils, water, air, and life. And it is critical in the sense that its functioning is very important for the sustainability of human life and all other life forms of Earth. And it plays unique functions and even ecosystemic services in terms of water quality and food production and regulation of waste. And I quite like this approach of the critical zone. And my own focus is within hotspots of the critical zone, such as the soil-plant root interface in continental terrestrial ecosystems, the sediment-water interface, and other interfaces. I will now move to the next slide, number 13, which illustrates the concept of critical zone biological functionings at these specific interfaces. On the bottom side of the slide, you can see specific interfaces. For instance, drilosphere, which is a part of soil that is directly affected by earthworms activity, basically the holes that are drilled through biogeochemical activity of earthworms. We also have microniches of organic matter, which is a mosaic of small-scale microhabitat dominated by several microorganisms such as bacteria. And last but not least, we have also the rhizosphere, which is a few millimeters of soil that is directly attached to the plant roots. And in this rhizosphere, you have a complex interaction between bacteria, plant root activity, such as secretion of small acids known as exudates, and also fungi. And if you want to understand the global cycling of trace metals within watersheds, even at the larger scales within the critical zones, you need to assess and investigate small-scale processes that occur in these unique hotspots, the drilosphere, the rhizosphere, and the organic matter microniches. In fact, locally, the physico-chemical condition might be very different from the bulk soil conditions. For instance, in a given anoxic or globally anoxic sediments, when waterworm activity by bioturbation or bioaggregation introduces some oxygen locally, therefore the condition will change even at a very small scales. And in this case, I'm talking about millimeter scales heterogeneities in sediments. Again, if we compare the bulk soil from the rhizospheric soil, we may find an enrichment in organic matter, in microbial activity directly attached to the plant roots. And this will be very different, of course, from the bulk soil, from the nude soil, from the soil uncovered by plant. So basically, my fundamentals is to understand these small-scale processes occurring at the interface between organic matter particles, mineral particles, solutes, and microorganisms to better assess what's going on at a global scale, at a higher integration scales. And therefore, you need a unique expertise in microbiology because the driver are the microorganisms. As I say to my students, the best chemists in the environment are for sure the bacteria. You have to understand in soils at least the activity of the mesofauna. And I also say to my civil engineering students that the best engineers in the soils are the earthworms. They are able to dig and drill and eat a huge amount of soils. And without these earthworms, soils and even agricultural soils would be much different. And last but not least, when you have this heterogeneous distribution in soils or at the sediment-water interface of microorganisms together with the fuel sources of organic matter, as fecal pellets, for instance, you also need to take into account this activity. Therefore, surface environment, soils, sediments are a mosaic of extremely varied sources of organic matter, of microorganisms with an interplay with the mineral cycle of elements together with the minerals themselves. We now move to the next slide, which illustrate what we are doing in our team, my own team. It is a geomicrobiology team where we try to understand what's going on, for instance, in the interface between soils, organic matter, minerals of soils, and plant roots activity that also hold specific microorganisms such as fungi and bacteria, not talking also about direct physiological activity of plants through, for instance, the secretion of exudates. And these biochemical factors, these physical factors, are very important in understanding the speciation of trace elements. And by speciation of trace elements, I do mean the physical and chemical form of the metals, so metalloids. For instance, if they are occurring as a free ion, if they are occurring as a complex species with dissolved organic matter, if they are adsorbed or sorbed on minerals, or if they are co-precipitated with some other major cations on the surface of organic matter, solid organic matter, or even minerals. And I do take speciation of trace metals in a broader sense than the union of applied and pure chemistry definition, because I also mean that the physical form of the metal or metal is also very important. For instance, you can discriminate between colloids, true dissolved metals and metalloids, and metals that are associated to particulate matter. I will now move to the next slide. It's about trace metal cycling and the environment, and maybe you can see it already. It shows that in our environment, in contrast to organic contaminants that can be always destroyed or may decay in the form of the basic form of carbon, hydrogen, oxygen, nitrogen, metals and metalloids can never be removed. They always change their physical and chemical forms. They are always cycles or immobilized, though this immobilization is also sometimes short-term, depending on the environmental control factors. And by these factors, I mean the physical variables, for instance, the amount and concentration of particulate matter in a river, for instance, chemical variables, for instance, the pH of the soil or even the redox status of the sediment, And last but not least, biochemical and biological variables such as plant activity, plant physiology, microbial activity, and also fungal activity. Not talking also about the mesofauna activity in terms of bioautomation or other mechanical aspects, but also chemical induced changes, for instance, through the physiological activity of earthworms. And therefore, the physical and chemical speciation of metals will influence their reactivity, their remerization patterns, and also their mobility and their environments. Other factors, such as biological factors, will then also influence, from the point of view of the receptor, its ecodynamics and their ecotoxicity. And therefore, mobility, transport, bioavailability, and bioaccumulation will be controlled by both biological and physicochemical variables. I will now move to the next slide. Here you can just take, maybe you can see it now. You have at the left-hand side of the slide the Mendeleev table showing which trace metal or metalloid is toxic, which one is a nutrient, and which one is both toxic or nutrient depending on its chemical form, but of course of its total concentration. So therefore, the speciation, reactivity, and transfer of trace metal in this critical zone is very dependent on key processes that are illustrated in the right-hand side of the slide, where you can see different forms of metals. For instance, in blue, you have the free ions, and this is as the interplay between several phases: the colloidal phase, the particulate phase, all particles for us geochemists higher than 0.45 microns, and the truly dissolved phase. And in the particulate phase, these metals, trace metals, may adsorb or even co-precipitate with particles, whether inorganic or organic, or even be adsorbed or absorbed in biological cells. As truly dissolved metals, they might be complex with inorganic elements such as salts, or also complex with small organic ligands such as small acids that may occur as root exudates in the rhizosphere. And last but not least, in this intermediate phase, which is lower than 0.45 micron and possibly higher than a few kilodaltons, you may find the colloidal fraction where humics, at least in soil poor water, play a key role in complexing the metals. That is, soil organic matter is also very important in the preferential transfer and mobility of trace elements. I will now move to the next slide. This is maybe you can see the title of the slide is Origin and Biophysical Control Factors of Trace Metal Hotspots. And the main thrust of my fundamental research is to understand the origin of these microniches, of these trace metal hotspots, and also understand how several biophysical control factors may affect their occurrence and diversity. For instance, the distribution of organic matter, the source and type of organic matter, physicochemical control factors such as pH and redox status, but also microbial or biological activity. And here you can see on the right-hand side of the slide some heterogeneous distribution of fecal pellets at the sediment-water interface. Basically, you have post-toxic or even anoxic sediment, but because of the hole just drilled by waterworm, let's call it waterworm, due to mechanical bioturbation, some oxygen might be introduced in the globally anoxic sediment, and therefore the local redox statue will change, and therefore the speciation, for instance, of uranium-4 occurring as uranium oxide may change, and it will be further oxidized with this newly brought oxygen into uranium-6, uranyl ion, and therefore become very mobile from one reduced form occurring as oxide to one oxidized form occurring as ion. And the different scientific objects I have been working on are the sediment-water interfaces. For instance, it is what you can see now on a new slide, number 18, which is the scientific objects. Fecal pellets, due to the heterogeneous distribution of organic matter that is settling and sedimenting at the water-sediment interface. The focus of today will be the rhizosphere, which is number B, the few millimeters of soil that are directly attached to the plant roots, and depending on the plant type, species, varieties, cultivars, and the plant phylogecal activity, you may find a very small rhizospheric system as for shrubs, as for herbs, but also very large one as for some trees like poplars and so on. And then you have in soil also drilosphere, which is a part of soil that is directly affected by earthworm bioturbation, and you may find at least 3 different types of physiological activity for earthworms: those who stay below ground, those who go up and down, and those who stay above ground. And last but not least, often sediment-water interfaces are covered by very thin biological systems known as biofilms, which are consortiums of bacteria and other microorganisms. And in these biofilms, in this interfacial system, you may find some very localized concentration of nutrients, dissolved gases, and even metals and metalloids, which is completely different from the bulk sediment. It is, in fact, a transition system. And understanding how these systems works will help us to better understand the global cycling of metals, better design the nature-based restoration system of contaminated media, and also better assess the fluxes of metal from one environmental compartment to the other, for instance, from the soil to the groundwater, from the sediment to the water column, and so on. I will now move to the next slide. Some approaches will be used for all these studies. Maybe you can see slide number 19, which is called methodologies. So we do some field work, some field measurements, some field observations, some field sampling. But this approach is also consolidated by laboratory experiments in terms of mesocourse experiments. And here in the central photograph, you can see some mesocourse of water sediment interfaces where you can control, in contrast to field studies, all the conditions: temperature, light, oxygen content, and so on. And last but not least, and you can see this in photograph on the right-hand side of the PowerPoint, you may also use simplified system, for instance, soil columns with several mineralogical phases, so simplified system where we try to understand the fundamental processes. And last but not least, we also try to combine these experimental approaches together with geochemical modeling, trying to understand what's going on in the field, in the laboratory mesocourse, or in the simplified laboratory system, such as columns. And we use several geochemical modeling codes to understand the reactivity of trace elements together with organic matter and minerals and living species such as microorganisms. I will now move to the next slide. Okay, maybe I skip that one. I will move to the slide number 21, which is about the objectives of the talk of today. Maybe you can already see it. It's about soil metal plant ecodynamics. So in the soil metal plant system, several biochemical factors are very important, such as organic matter, soil organic matter, but also in the rhizospheric system, root exudates as small acids that are excreted by plant roots. Then you also have biological activity in terms of microorganisms, fungi and bacteria alike, that are hosted in the plant roots. That is very important to understand the speciation and their role into the translocation of metals from the soil to the soil pore water, to the plant root, to the rhizosphere, and to the upper organs of the plant. And therefore, in natural systems, you find a competition between trapping phases such as soil organic matter and soil minerals, together with plant root and rhizosphere and plant activity. I will now move to the next slide. I skip slide number 23. I move direct to slide 23. The slide is called Essence of Phytoremediation. Of course, soil pollution, as foreseen by the World United Nations Food and Agriculture Organization, soil pollution is a hidden reality. Anyway, we can take care of it if we understand the fundamental processes in the soil plant metal systems. And for metal contaminated sites, it is possible to design some remediation strategies based on plants. It is called phytoremediation. And so far, we have mainly worked on phytostabilization, with the use of higher plants, higher organisms to stabilize metal and metalloid contamination in highly contaminated soils, such as mine soils, technosoils, and post-smelting area soils. However, it is also possible to design for less contaminated soils some remediation strategy based on phytoaccumulation. And phytoaccumulation is the use of specific plants that may concentrate in the upper organs, metals and metalloids, to a great extent. And at least in France, several plants, such as shrubs or even herbs, have been identified in former mining areas. I will now move to the next slide. In order to understand, optimize, rationalize, and design new phytoremediation strategies, it is very important to understand what's going on in the rhizosphere of the soil plant metal system in terms of rhizospheric processes. Of course, the rhizosphere is an interface between the bulk soil and the plant, and it presents many physical, chemical, and even microbiological gradients. Here on slide 24, you can see how several chemicals, several compounds are exchanged between the plant root and the bulk soil. For instance, you have, besides an uptake of water and mineral elements related to the water flux, an influx of oxygen and an efflux of CO2 that may locally alter the pH of the rhizosphere. You may also have an absorption of major mineral elements, so for instance, calcium and magnesium, that may compete with some of metals and metalloids in the rhizosphere, and the bioavailability to plants has to be understood at the level of these rhizospheric processes. And therefore, we developed several experimental approaches to be able to understand the speciation, reactivity, mobility, and translocation of metals and metalloids in the plant root soil rhizospheric soil system. I will now move to next slide. Maybe you can see it. It's slide 25. It's called Triggers of Bio and Phytoremediation. And here I have classified plants according to their tolerance to metals. You may have some non-accumulator plants, some even excluder plants, that is metals and metalloids will be excluded and stored in the plant roots. And this will be excellent for phytostabilization. You may have some mildly accumulator or even hyper-accumulator plants, and this will be very interesting for phyto extraction. And of course, whereas for phytostabilization or phyto extraction, The key thing is to understand what's going on in the plant root systems, because bioremediation is often combined with phytoremediation in the sense that microorganisms that are hosted in the plant root systems, fungi and bacteria, these are the ones who do the tricks prior to possible translocation of metallic contaminants in higher organs of the plants. Okay. This is what I mean by rhizospheric effect, a collective effect of root exudates related to plant activity, soil root exchanges, but also microbial activity in the rhizosphere, in the plant roots, from fungi and bacteria alike. I will now move to the next slide. It presents the specific objective of our study. And basically, we did some pot experiments in the laboratory with some contaminated soils taken from various parts of France. And these soils were also amended with biochar being a residue of organic matter that is degraded and burned in reducing conditions, that is producing an amendment that is very useful to increase the soil fertility. But recently, this biochar has been also used for the remediation of contaminated soils, of metal and metal-rich contaminated soils. And when combined with phytoremediation, it is also very useful. And we are talking about assisted or aided phytoremediation, aided phytostabilization, even aided phytoextraction. And in these experiments, we did work with different biological models, salix and lolium, which is basically trees of very common in France, and also some herbs very common in France. I will show you some picture effects through the development of these models through time in pots from contaminated soils, and focusing really on the rhizospheric effects, that is root exudates, microbiological communities development and their activity, and their interplay with the ecodynamics of trace elements, metals and metalloids, together with their speciation and the changes of control factors, pH, oxidological potentials, and so on. I will now move to the next slide. Maybe I don't have time to go into much details. This shows how we interrelate all our methodological approaches from the different studied level, from the bulk soil to the specific gene of the microbial and fungal communities. So if we take the bulk soil, basically we do some bulk characterization in terms of compartmentation of trace elements, in terms of bioavailability and fate of trace elements as a speciation, doing some standard chemical measurements in terms of pH, redox potential, conductivity, dissolved organic carbon, and so on. Then we also do some partitioning at the scale of individual minerals that are very important in terms of sources, because they are carrier phases for these contaminants. And we are doing some electronic microscopy, some chemical sequential extraction of metals from these phases, together with infrared spectroscopy and X-ray diffraction for mineralogical characterization. Plants are cultivated under controlled conditions, and at the end of the experiment, these plants are sacrificed, and individual organs, such as stems, roots, even leaves, are digested to investigate the mineral content, the mineral mass in terms of metals and metalloids. And then within the rhizosphere, within the rhizospheric soil, we also do some analysis of this specific soil attached directly to the root systems, together with geochemistry in terms of root exudates. And the next steps will be to investigate the microbial community that is directly attached to the plant roots in terms of fungi and bacteria. And we use all kind of proxies, all kind of biochemical analysis such as enzymatic activities, some molecular ecology techniques based on DNA extraction and so on. I'm not very familiar with that, but I will collaborate with molecular biologists of the nearby French Geological Survey for that. They're into PCR and all kind of analysis after sequencing, and I find this very interesting. And the next step, the last step, will be to try to identify specific gene, for instance, for arsenic oxidation of arsenic reduction of specific bacteria in the plant root systems. So from the bulk soil to the microorganisms, we need to understand everything, all the compartments, all the actors, and therefore we will be able to rationalize, optimize, and design new remediation strategies based on assisted phytostabilization. I will now move to the next slide. This is section number 4, material and method. You have the geological map of France. And if you see in the very center of the map, you have some red areas that correspond to very old Primarian rocks. And in this part of France, many mines have been operated. They are, of course, now closed from the Roman times. Gold mines, silver mines, lead zinc mines, and they have been untouched from the years 1940 to 50s. However, many tailings, many waste have been there, and new soils have developed on these tailings, and they are very enriched in metals and metalloids, for instance, cadmium, lead, zinc, copper, and metalloids such as arsenic and antimony. And these new developed soils have been developed on tailings, have been colonized by vegetation. So these are unique laboratories for investigating the interaction between plants, soils, and metals and metalloids. And if we can learn what's going on in the field, if we can validate what's going on in the laboratory in simplified experiments and backed up by geochemical modeling, we are then able to select some specific plants, select some specific amendments, such as biochar or even compost, and try to diversify the panel of biotechnologies that may be useful for remediation, at least as a phytostabilization of metals and metalloids in other contexts, from the mining context to industrial or post-industrial context in other parts of France. I will now move to the next slide. This is a Rhizobox experiment from soil that were sampled in the north part of France at the border with Belgium. The site is called Mortagne du Nord. Maybe you can now see the slide. It is called Rhizobox experiment. And in these former smelting area soils, highly contaminated with cadmium, lead, and zinc, we did some experiments with poplars, with cuttings that was directly implemented in the soil. And we divided our boxes into 3 parts. The rhizospheric soil that was roughly divided and separated from the bulk soil with some mesh. And then we also allow these cuttings to grow. And through time, sampling of soil pore water was performed in order to assess the evolution of the total concentration of cadmium, lead, and zinc, together with the speciation and together with control factors such as pH, redox status, but of course the total organic carbon, dissolved organic carbon content. This is a concept of Rhizobox experiments where you try to simplify what's going on in the lab, and we are using this model of poplar, Populus americana dorscams. And poplar is interesting because it has a high biomass. It has a mildly accumulating capability for heavy metals, and it also develop important root system above ground. So therefore, it can explore larger portion of plants, of soils compared to other plant species. So now you should be able to see slide number 30, which is about the investigated parameters. You can see a focus on the Rhizobox. So we did monitor the pH of the soil pore water in the rhizospheric part of the box, dissolved organic carbon, together with the total dissolved concentration of potentially toxic trace elements, in this case cadmium, lead, and zinc. And at the end of the experiment, all plants were sacrificed, the different part organs were separated, and the total content of metals in the different parts of the plant organs were determined, together with the dry rate production of the different organs, in order to understand the potential translocation from the bulk soil to the rhizospheric soil, to the plant root, to the upper organs of the plant, and assess the potential of this poplar, of this specific cultivar for bio remediation, for assisted phytostabilization. I will now move to the next slide. Here are some experiments, another experiment carried out with a former silver mining site, where you have similar contaminants as in the previous experiments, together with arsenic and antimony also. So heavy metals, zinc, cadmium, lead, together with metalloids such as arsenic and antimony. And here you can see the effect of the biochar amendment. Maybe you can see the Salix vinimalis experiment, which is another tree, Salix, willows. And you can see that the addition of biochar up to 5% mass does increase the growth of our cuttings of a 5 weeks experiment. And it's also the case with another experiment with some herb grass, ryegrass, Latin name for the species being Lolium perenna. So in this other experiment, we wanted to investigate the effect of biochar on the speciation of trace elements, metals and metalloids, root exudates, and microbial community with these two biological models that may be used for phytostabilization or even for phytoextraction, as is the case of ryegrass. I will now move to the next slide to present you some more methods. So it should be slide 32 with investigative parameters. So on the left-hand side of the PowerPoint slide, you may see the willow cuttings that we have implemented in a pot experiments. And I want to see you how we sample actually the soil pore water. And this is done with some rhizome samplers developed by our colleagues at the University of Wageningen in the Netherlands in Europe, Holland. Basically, the idea is to use a depression, a small searing that is introduced nearby the plant root system to sample the soil pore water on a regular basis. And this filtering capacity is 0.20 micron, and therefore we do sample the dissolved species. We are able to do some chemical measurements such as pH, dissolved organic carbon content, but also this total dissolved metals and metalloids concentration. And here you can see an example of a willow cutting after 2 days, but also after a growth period of 55 days. So it has very much developed. And as I said before, at the end of the experiment, the organs above ground, below ground are sacrificed, mineralized, and the metal concentration in the different organs, roots, stems, leaves, are determined. I'll now move to some results, I think. I skip this one just to see you how we proceed with the experimental plan, the experimental design, control experiments, that is soil without plants, soil with different biological models such as Salix or ryegrass, and various amendments such as biochar at 0. This is another control, 2% or 5%. I shall now leave to slide number 36. It will appear anytime. It is a section 5 results. And here I want to present some results of the first soils I have talked about, the one at the border between France and Belgium in the north part of France, the former smelting site of Mortagne du Nord. And here you can see some key characteristics of these soils that have a very important heavy metal content, way below the local pedo-geochemical background, a quite important organic matter content. There are some pre-layer soils with a diffuse contamination of metals. They have an interesting nutrients balance and a slightly acidic pH. You have a concentration gradient from zone 1 to the zone 3, depending on the distance from the original smelting unit. I move now to the next slide for the result. And here, it is slide number 37, where I present soil pore water metal concentration in the bulk soils and in the rhizospheric soils. So for each graph on the left-hand side of the PowerPoint, you have the pH. Above that, you have another graph with the dissolved organic concentration in terms of milligram per liter. And on the right-hand side of the slide, you may have a total dissolved concentration of zinc, cadmium, and lead respectively. And you have two colors, the white one being for the rhizospheric soil solution, and the darker one, the gray one, being for the bulk soil solution. So from a statistical point of view, you may find some key differences in terms of physico-chemical characterization of the bulk soil solution and the rhizospheric soil solution, up to a few dozens of milligram per liter for dissolved organic carbon, up to a few 0.5 pH units for between bulk soil and rhizospheric soil. And this will also influence the total concentration of metal and metalloids, as locally you will have some changes in the pH, which will of course play on the precipitation and dissolution rates of zinc, cadmium, and lead, but also on the absorption with the mineral fraction of the soil. And last but not least, the presence of dissolved organic carbon will also play a key role in the complexation of zinc, for instance. And I will now move to the next slide. With this slide, okay, I skipped the slide 38. Let's move directly to slide 39. It should appear anytime. It is the title is concentration of metals in popular organs, and here you have the cumulated concentration, the mineral mass of zinc on the left-hand side of the slide, cadmium in the middle, and lead in the right for the different organs: leaves, stems, roots, and other upper parts of this poplar that has been grown in contaminated technosoil during 60 days. And basically everything that is below zero is the roots, and everything that is above ground is for the upper organs. And you can see specific trends. Lead seems to be all exclusively associated with the root systems, whereas zinc and cadmium, of course, they are more or less associated with the root system, but to some extent, there was some translocation in the upper organs. And this is not, we can well conceive this because zinc is also a nutrient at lower levels, such as copper, and therefore may be translocated from the root to the upper parts of the popular organs, such as leaves. And cadmium seems to have an intermediate role, and it may substitute to zinc, even compete with zinc. And cadmium, in contrast to zinc, does not play any biological function. It is a contaminant only. And organs, you may also assess if the plant is optimal for phytostabilization or even for phytoextraction for contaminated soils. I shall now now move to the next slide. Some results on the former mining soil of Pongimo. Remember the former silver mine that is very contaminated in lead, zinc, cadmium, but also arsenic and antimony. Here you have a picture of a pedagogical unit that was digged on the site. You can see that the texture of the soil is very coarse, that is mostly sandy. And if you take the table, which is the right-hand side of the PowerPoint slide, you may see that the total concentration of metals and metalloids is extremely high, way beyond the local geochemical background. But that also the soil is very poor, that the pH is much lower than the previous one, and also that its mineral content is low. So it's typical of technosoils developed on these mine tailings, low mineral content, lots of contaminants, metals and metalloids, low organic status, low mineral status, and low pH, and so on. Let's move now to some results for this former mine site. And let's see how it behaved with the plans we try to implement. You should now be able to see a graph with the title is Soil Power Water Control Factors. And here I do present pH and redox potential for the various experiments. I02, I5 as the control experiments. And you can see a clear effect of the biochar. By adding biochar, you strongly improve the pH status of the soil, because biochar is an alkaline material. And of course, the more you add it, the more the pH will be improved. And if we now consider W0 to W5, it's a willow experiment, and R0 to R5 is a raygrass experiment. And NV, 0 to NV5, is a non-vegetative experiment. It is a kind of control experiment with biochar, but without the plant roots. I will now move to, to allow these plants to develop on these very hostile soils, adding some organic amendments such as biochar may favor the development of a vegetal cover. I now move to the next slide, which is electrical conductivity. It is called, the title of the slide is Soil Power Water Control Factors 2. And you can see the effect of biochar amendment on the nutrient status of the soil, where you can see that electrical conductivity is pretty much improved, and also on the dissolved organic carbon of the soil power water, where this biochar amendment plays also very interesting effect. Last results, I now move to another slide. It is slide number 43, where I do present final results on the total dissolved concentration of metals and metalloids in the soil power waters of these technosoils amended with biochar and cultivated with raygrass or willow. You can see that for many heavy metals such as lead and even zinc, the concentration, total dissolved concentration is very much reduced, together with the addition of biochar and the cultivation up to 5 weeks of willow and raygrass. And it's also the case for metalloids such as arsenic. I do not present here any result for antimony, but in our system, antimony behaves quite similar to arsenic. And last but not least, for very potentially toxic elements such as cadmium, the effect was also very positive. And last but not least, if we now correlate the concentration of metals and metalloids in the soil power water together with the concentration in the different plant organs, it is slide number 44. We can see on the left-hand side of the slide some results for willow, and on the right-hand side of the slide some results for raygrass. And you can see for various conditions of non-vegetated soils with or without biochar additions, a translocation of or not of metals that follows previous study results. For instance, lead seems to be more or less stored in the root of the willow and the grass. It is also the same to some extent for cadmium or zinc. So, though some zinc and cadmium is translocated in the upper organs of the willow and grey grass, and arsenic also seems to be highly stabilized in the plant roots, as it seems to be excluded from the higher organs. So I should say that willow, but also raygrass, are able to develop on these mined soils that are able to phytostabilize heavy metals together with metalloids in very contaminated environments, and they may be able to be transplanted in other contexts, in industrial contexts, or even in less contaminated contexts such as urban soils, for phytostabilization options. I now move to the conclusion. It is slide number 6. You may see it anytime. Section number 6, conclusion. So, two conclusions on biochar. Biochar, which is a unique organic amendment that can be also very cheap and produced in large quantities, modifies soil physicochemical parameters and therefore influence directly or indirectly the distribution and availability of metals and metalloids. Nevertheless, it also increases plant growth by improving the nutrient status of the soil and also the pH status of the soil, which is very problematic for technosoils or mine soils. Another conclusion from the point of view of the plants, whereas raygrass, poplar, or willow, these plants have been shown to generate a rhizospheric effect via root exudates mainly, via small organic acids we are trying to investigate. And it will, this biological activity will modify locally physicochemical parameters in terms of pH and complexation of metals and metalloids, therefore modifying their bioavailability. And in turn, trace metal speciation, translocation, sequestration by high organs, and bioavailability can be improved. And we shall conclude that these plant models are very useful, or will be very useful when fully understood, when fully rationalized and optimized for biostabilization of metal and metalloids in highly contaminated soils. I would like to thank you, all of you, for your attention. I'll be ready now to answer your questions. And this is just a picture of our beautiful region of the River Loire, not far from here, showing the sandbanks of the river, which move from one season to the other. And I was very happy to present my work. I hope you did follow it. You will be able, thanks to Dr. Manash, to download all the slides. I had to skip some of them. And now I'm happy to answer your questions. Maybe I try to read them in the chat box, or maybe listen to you. Thank you so much.